plans of the MS Shawby without the luck cut around the people. You were at an event for the Department of Education. This is the C. Gordon Winder Lecture, a memorial lecture to Dr. Gordon Winder. He was a professor in our department, and he was interested in geologic science and focused on research areas of geologic stratigraphy, particularly sedimentary carbonate microfield development, urban development, which maybe some of us probably were doing. So he'd be pretty thrilled about today's topic. He was a strong advocate for earth science education in Oregon and chose to fund this lectureship called the SPUGOSH because he wanted it to be anonymous, but he was providing those funds for this event. It was to ensure a prominent earth scientist, preferably one that was Canadian, would visit Western semi-annually to provide an earth science talk that should attract the general public. And the lectureship began in 1999, 27 years ago. We're coming up on the 30th anniversary soon. And it has now been renamed the C. Gordon Winder Lecture. I would like to acknowledge that we are joined today by the daughter of Gordon Winder, Nancy Holmes. Who's giving the talk this year? It's going to be Dr. Marie Gingras. He's a professor at the University of Alberta and also an inaugural chair, also convinced to be the chair of the Earth and Atmospheric Science Department at U of A. He completed both his BSc and immediately went into a PhD geology degree at the U of A in the 1990s. And then went on to be an assistant professor at the University of New Brunswick for a short time before U of A snapped him back. Dr. Gingras' research focuses on using sedimentology, which I think this crowd knows, is the study of sedimentary rocks, like sand, floods, and pebbles. And also ichnology, which is the study of tree fossils, the footprints, burrows, and tracks of ancient animal, which will be pretty interesting to hear about. And the whole goal really is usually to understand and interpret ancient sedimentary environments. And you should know that has applications to understanding our modern sedimentary environments, paleoclimatology, and the hydrocarbon industry, which Dr. Murray Gingras worked as a petroleum geologist for some time. So let's welcome Dr. Murray to present to us about the practical power of ichnology.
All right, thank you all for coming. I have to say it's a real pleasure to be here and an honor to be part of the C. Gordon Winder lecture series. And, you know, it's a lecture that's in honor of academic effort, but more importantly, meaningful teaching to students and the public. And you know, Winder was a philanthropist as well, as is evident from the lecture, the existence of this lecture series. And, you know, I was really tickled to be asked to be a part of this. And you might not be aware of it, but there's some notoriety across Canada and geoscience departments with this lecture. And I had no less than 3 emails after Alina had circulated it to a few of the departments that she thought to share it with. And at least, well, all of them were like, oh, so you're a big shot now. You're a part of the Winder series. And I'm like, yeah, that's me, a big shot. Yeah, for sure. Yeah, it only took me 30 years. So it is an honor to be here. And I hope to take full advantage of it by giving you an opportunity to learn just some basic information or knowledge about a field that I trained in for my PhD primarily, which was ichnology. And ichnology, I think, is broadly understood in Canadian geosciences by sedimentary geologists. We're one of the what would you call it, scholarly cultures that get the importance of ichnological data. And you know, I think the very simple ways technology can be used are often overlooked. And I think that people who don't dabble in ichnology will appreciate the opportunity to understand kind of the elegance of the interpretation that ichnological data can drive into your overall sedimentary data. Now, what talk would be, well, maybe I got to point at the computer, try this, different buttons. Oh, there it is. You can't really, I think, give a general lecture about ichnology without, I think, talking about the contribution of two mentors of mine. One was my PhD supervisor, S. George Pemberton, and Adolf Seilacher, Dolph Seilacher, he preferred to be called. Seilacher worked at the Senckenberg Institute and was a co-appointee to Yale for many, many years. And George Pemberton, of course, primarily worked at the University of Alberta with some experiences at the University of Georgia and the Alberta Geological Survey before that. But the bulk of his career was occupied at U of A. And I would say that these two mostly are responsible for the foundations of what we would view as a modern framework for ichnological interpretation and how it can be used to bend the sedimentary record to your will. Adolf Seilacher, Dolph, did his work on his PhD in the 50s in Fliess in Europe, looking at ichnological distributions and ultimately deriving something that we'll get to later in the talk, if ever so briefly, and that's the ichnofacies concept. And Pemberton, who did his PhD in the 70s, really took the ichnofacies framework and extended it into this world of the late 70s and then into the 80s of facies models, which for those of you who aren't that familiar, was largely led out of McMaster University, where George did his PhD. And the McMaster work was really led, the facies modeling, by Middleton and Walker, and George's position in ichnology and sedimentary geology at the time kind of made him the crucible of what really became modern ichnological analysis, fundamentally the framework that almost everybody follows today. And I've spent my life in ichnology, adding precious little compared to what either of these men have done. I will point out, and not to embarrass Josh, but George's son, Josh Pemberton's in the crowd at the back here. He's a professor here at Western, and I think a great asset for you. So what is ichnology? I don't know. The simple answer is it's tracks and trails, in general, fossil tracks and trails, and burrows, right? And mostly what we're looking at in the rock record are these infaunalizations, or animals living in sediment. So these might be tubes and tunnels, whatnot, but it includes little trackways and so forth. And they are used to interpret, I do have this, paleoecological and depositional data. And I say interpret, they add that to a sedimentary interpretation. And, one really great thing you'd say about trace fossil work is it adds a lot of data without spending a lot of money on it. You don't have to mill the rock and powder it and put it through an ICP-MS. You don't have to use XRD. You're adding data that's functionally free except for your time looking at the core. And if you're admiring this wonderful bit of core that we're looking at here, this is the McMurray Formation. And I was working on a project with Amoco back in, it was actually BP. Amoco bought them out that year, or was it vice versa? Yeah, it was BP bought Amoco out. And I was mapping for them for the McMurray Formation, and we were logging all of this core. And of course, this one is the perfect ichnological core because that's a trace fossil that says everything you really need to know about ichnology. And trace fossils in of themselves have a history in geoscience that's a little bit complicated by the fact that they were initially interpreted as seaweed. So they were called fucoids, and the taxonomy of many trace fossils goes all the way back to the late 1800s when they were treated as the fossils of marine algae. And when you look at this trace fossil that's called chondrites, and it's weathered out rather impressively, you can kind of get the sense of why you might think of that as a fossil seaweed. And in fact, the name chondrites and other names of trace fossils like Cruziana are all derivative of their original misinterpretation. However, by the time we get to the, I don't know, probably between 1890 and 1920 was really a revolution in the thinking of trace fossils. And we get to this point where it's pretty well understood that they're not just biogenic sedimentary structures, but they're biogenic sedimentary structures made by the activities of animals moving in or on sediment. And on the left, you see what, and I'll give you full disclosure here, that's not a real picture of a trilobite. I used AI to do that. And like everything AI, you get a pretty mixed result, right? That's supposed to be the trace fossil Cruziana, and it's not. But it's a good enough proxy for this presentation. And I wanted something that contrasted heavily with the other Ying. This is a real picture, the vertebrate tracks. And these are both trace fossils. One is easy for the human mind to interpret, trackways. We make our own trackways that aren't overly dissimilar from whatever this hadrosaur made. And then on the left, the idea of interpreting invertebrate animal trackways and burrows becomes a little more abstract. Other trace fossils might include tubes that worms live in. This is from a modern study area. It's from my PhD, actually. Probably took that photo in 96. And it's at Willapa Bay, which was my study area. That's a little estuary on the west coast of North America in Washington. And George had me working on that in concert with some former USGS employees who were interested in integrating a strong modern sedimentological record with the ancient, there's a Pleistocene set of terraces that are the fossil or ancestral Willapa Bay, and so they could be contrasted. But I love these tubes. It's a little forest, literal a forest of worm tubes that are made by the polychaete Maldanus maldanus. And we use ichnology, modern ichnology, we call it neo-ichnology, as a prism through which to interpret some aspects of the ancient rock, of the rock record. It doesn't work all the time, right? Because, for example, things that were made by Cambrian animals may not have that many direct analogs anymore. But by the time you get to the Triassic, things are looking pretty good. And into the Cretaceous, we're looking at a world that resonates quite well with the modern. There's traces that aren't well studied at all. This fella here, Walter Schinkel, has actually studied the nests of primarily ants, but not solely ants, since the 90s. I think the first article I saw from him was from Popular Mechanics, of all places, and Discovery magazine, both almost at the same time. Discovery magazine doesn't exist anymore, but it was certainly a favorite of mine. And this is a plaster cast he made of an ant nest, which is unbelievable in its completeness and recovery. A few years later, he started realizing that you could use molten zinc to make high fidelity casts of these ant nests and get really good models of them. So he wrote this whole book called Ant Architecture, showing all these architectures. Now, he assures the reader in the book that these are all abandoned ant nests. And so that's what Walter says. But I must admit, I love the architecture. I mean, these look like space stations, right? This is where ants live. And it is fascinating that, you know, in the world of trace fossils, it's fascinating how little attention insects, land-dwelling arthropods have really received. But let's go back in time to when bioturbation appears, and that's the Ediacaran, okay? We don't see trace fossils till the Ediacaran, so there's nothing in the Neoproterozoic before the Ediacaran, although there are Ediacaran fauna that appear before bioturbation. Bioturbation comes a little bit later. And that bioturbation is generally pretty straightforward, right? It's a set of simple traces that are linear to curvy. And, you know, there's no infaunalization, although there's at least one published study that shows infaunalization in Ediacaran, but there is only one or two. What, you know, digging in sediment, that's what I mean when we say infaunalization. So there, you know, there wasn't necessarily much going on in Ediacaran, but the oldest trace fossil we know of is a contentious one. It's from the Takuari Formation. Not everyone agrees it's the age it is, and it's 583 million years old. And it's really consists of these simple furrows moving through the sediment. What was it dated on? This is a sandstone, right? And these simple furrows are, it was dated by cross-cutting dikes that are that age. So the 583 million years is actually a minimum age, right? So that fits the time frame of molecular clocks pretty well, where we think that the ancestors of animals are erupting somewhere around 600 million years ago. But molecular clocks are not that great for telling time back then. And so there's a lot of uncertainty in that. If we ignored this contentious example, the next known trace fossil is only 555 million years old. That's in the White Sea fauna of Russia. And it's already a kind of a complex trace fossil in that it really has a central burrow and an animal that was scraping food into that burrow like a slug or something like that with radular teeth. So there would definitely bioturbation before the White Sea fauna, and I would contend that this is at least a candidate. And it's hard to get by the cross-cutting dikes, but let's leave it at that. What was the first animal like? And the first animal was, no, okay. The first animal is actually only a matter of speculation. No, I mean, we know something existed. We call the ancestor of all the animals Ubilateria. So that's the name for this unknown animal. And what do we know about Ubilateria? Well, it had to be simple. It was probably vaguely slug-like. Whether it was a fat, short little slug or a long, skinny little slug is a good question. And, you know, to have the characteristics of later animals, it really needed a mouth, a digestive tract, some way of sensing its environment, and some way of moving about. So this could have a bilateral symmetry because it's elongate and it has some musculature. And that's how you would explain the bilobate symmetry of that tract. So that's Ubilateria. And Ubilateria has never been discovered, although in the, what is it called in China, the Qiangxi fauna, they have candidates for that, very teeny candidates, and not everyone agrees with that. By the time you get to the Phanerozoic, you're dealing with trace fossils that are decidedly more aesthetic. And this is all the way up in the Ordovician, and I only chose it as an example because I just love the patterns that are made by the animal on the sediment surface. So this isn't infaunalized. And by the Ordovician, we have lots of infaunal burrows, but we're not going to go too into that. The explosion in bioturbation happens in the Cambrian. Guess when? Well, about the time of the Cambrian explosion, right? That makes perfect sense that at the Cambrian explosion, you get this proliferation of body forms, and this proliferation of body forms is expressed as trace fossils. And I would argue, based on the trace fossil record, that the explosion in trace fossils actually precedes the explosion in body fossils. That could be taphonomy. But, you know, it's a good question. Does behavior, which is what trace fossils are, and we're going to get to that, is that the tail that wags the dog, or is body form the tail? In other words, does body form dictate behavior, or does behavior predict body form outcome? And it's actually an interesting evolutionary question, one we don't have to answer here. These were likely made by trilobites, but it could have been other arthropods. You get up to the modern, and we can study trace fossils really intensely from the Cambrian all the way up to the modern. And the left-hand pictures from my PhD study It's a sand shrimp called Neotripea californiensis, making tunnels in the sand. And I was studying the behavior in these thin-walled aquaria at Willapa Bay. And then on the right is from Cooking Lake, Alberta, right by Edmonton. I had me and a PhD candidate or student. He is a candidate. Ryusuke Kimitsuki were establishing the freshwater fauna, which are way different than the marginal marine fauna. And so this is one of the pictures from his study that was just recently published. And so you can kind of bridge this world from the modern to the ancient. And the fun thing about working is in ichnology is spatially you can work anywhere. Like I work in estuaries in the modern, I work on shorelines and bays all over the world, but I also work all over the rock record from the Precambrian all the way up to the Pleistocene, right? So the ichnologist's diet is much more diverse than many geoscientists get to enjoy. So why would we use ichnology? And I think the most important point is right number one there, which is trace fossils are in place. Right? Body fossils can always be transported. They can be reworked. They can be reincorporated. And sometimes even if you have, let's say, a fully articulated specimen of a vertebrate animal, you don't know it actually died there. There's always some question. And with trace fossils, these are disruptions in the sediment that dictate that show that the animal was living there. In other words, that presence of the animal and its activities can be used to directly interpret the sedimentary conditions at the time that bioturbation occurred. And they occur in many different sedimentary environments. This changes through time. For example, in the Ediacaran, you mostly have trace fossils limited to maybe the lower shore face, which is near the shoreline, to the inner shelf. There's maybe some exceptions, but that's what I would argue. And then by the Cambrian, you're starting to get broader trace fossil distributions on the shelves and shorelines. By the time you get to, let's say, the Permian, although there's examples before then, that you start getting bioturbation profusely on land. But the first trace fossils on land actually appear in the Silurian, and there's some animal walking across a dune face, and it's actually from Ontario. The first land trace fossil was reported from here. And finally, anyone who's a paleontologist knows that there's limitations to body fossil preservation, pH conditions and compositional differences in water or compositional characteristics of groundwater in general. The oxidative qualities can lead to the complete dissolution, disappearance of a body fossil. But because a trace fossil is a textural expression of the sediment, you don't lose that. And I like this. This example's fine. This is a Skelethos it's a vertical trace. An animal lived in here, like a worm hanging out there vertically. Nice, simple trace fossil. But you can see that there's a compositional difference between the burrow fill and what's surrounding it. And as such, it would take quite a degree of metamorphism to actually lose your ability to interpret that trace fossil. So as long as there's textural heterogeneity, you're kind of set on preserving your trace fossils. And what you have is a long record of bioturbation, meaning most of the Phanerozoic has bioturbation in it. And by the time you get to the Middle Cambrian, your expression of the ocean shelves and shorelines, for example, doesn't change that much all the way to the modern, right? But you do have these radiations onto land. And the final point is that specific traces, often form, I don't know about often, under certain environmental conditions. So there are a few trace fossils that really help paint the picture of what bioturbation, that really specifically inform us of the sedimentary environment. And the most important reason to study ichnology from the rock record is that it contains the record of what we would have called in the 80s the minor phyla, the animals that aren't well represented in the fossil record, soft-bodied organisms. You don't see much in the way of fossil polychaetes. You don't see much in the way of fossil, well, all of these guys, sipunculids, anemones, all of these types of animals represent a huge proportion of oceanic biomass and an absolutely trivial proportion of the sedimentary rock record, fossil record, but their traces are proliferate in sedimentary rocks. And as such, they give us a key for decoding seafloor animal diversity through time, and they're used for that. I don't want to talk too much about trace fossil classification, but you should know that trace fossils are actually given these binary names. So it's a lot like Linnaean classification, right? You have in Linnaean classification, kingdom, phylum, class, order, family, genus, species, you have the genus, species, and that's how we usually refer to animals, by their genus and species, like Homo erectus or whatever you have. This is an ichnogenus, and this is an ichnospecies. So ichnogenus and ichnospecies are deployed pretty much the same way as Linnaean nomenclature, but there's not the long tree. Sometimes we have ichno-families, which is the next higher level, but for the most part, ichnological classification systems don't really bother with that. And the reason is that these are not grouped together like animals are or plants in an evolutionary tree, right? The trace fossils are named for their morphology, and so they have no true genetic relationship. So there's a limit to how far you really should be connecting these. They are, ironically, dictated by the zoological code, the code of zoological nomenclature, as to how they're named and how they can be named and who can name them and so on. And that means that trace fossil names are limited to biogenic sedimentary structures that were made by animals. Okay, if it's not made by animals, you can't name it an ichnogenus, an ichnospecies, which causes problems for ichnologists sometimes, because, for example, a plant root in sediment is for sure a biogenic sedimentary structure, but it has no name. We just call them like rhizoliths or something like that. And that's not the only example. Ichnology, Bill Sargent, a Canadian ichnologist, or a British ichnologist who worked in Canada for most of his career, became a Canadian, worked in Saskatchewan for most of his career, and actually wrote a code of ichnological nomenclature that was clear, straightforward, made sense, and never adopted by the community. Yeah, right? Isn't that the way it goes? Yeah, so just being a visionary isn't always enough. Some basic things about ichnology. And the first one I like to stop in its tracks. You know, if we were at a core lab and I showed you a trace fossil or something like that, almost the first question you're inclined to ask, and I understand it, is, well, what kind of animal made it, right? And that's actually a bit of a problem, because very different animals can make very similar biogenic sedimentary structures, which is a clear limitation in making your interpretation about who did it and how, right? There's different ways to make these structures. There are trace fossils that lend themselves pretty well to at least a general identification of the animal. But these vertical tubes like Skelethos really aren't. And on the left is some fanciful arthropod living in this shaft. In the modern, these would be like a little isopod. They live in burrows like that all the time. Or you might have a little worm, right? A little annelid. And so these are both the same trace fossil, and they are both reasonable candidates for interpreting that biogenic sedimentary structure. And I wanted, just because we were on the topic of ichnotaxonomy, to show you This here, this is an example of a trace fossil. And these are the types of morphological characteristics we're looking for. An ichnologist would refer to this as the causative tube. If a worm made this, say, that's the tunnel the worm was living in. That's all. And then your eye is drawn to these curved features in between the tubes. And we would call those sprida. And you can see the schematic. This is a picture from my PhD candidate, Arzu Accacelli, and she's working in the Imperial Formation of which Devonian age in the Mackenzie Mountains in the Northwest Territories. And she's made herself into quite a taxonomist, I have to admit. And we would call these sprida. What are those sprida? Well, if you could imagine that the end of the burrow used to be here, and over time, the animal kept moving the end of its burrow out, then what's left in its wake are these little curved shape laminations, which we call sprida. And so that's a record of the animal extending its burrow over time, and it might be over weeks, months, or years, right? Why would an animal do that? Well, maybe as a deposit feeder, right? And maybe it's mining the sediment for food, which isn't at all unusual. This trace fossil is not called Cruziana. That's cut and paste from the next slide, and then I couldn't fix it on this computer here. It's called Rusophycus. But what does Cruziana look like? Like that. This is a trace fossil that really is often ascribed to trilobites. So that's the one where with the trace maker, we feel pretty good about that being trilobites, but there are other arthropods that for sure could make Cruziana. But you can see it's got some features. It's got this furrow down the middle. It's got these scratch marks on each side. You can imagine those trilobites, they look like flat little bugs and they have those legs on the side. Well, they're in the sediment. They're using those legs to pull up food into their mouth parts and move along. They're deposit feeding. And so that's a really typical trace fossil called Cruziana. And we'll just flip the rule I gave you 3 slides ago on its head, which is that, okay, one needs to know that different trace makers can make similar trace fossils. We already agreed to that. And that's shown on the left here. This is an old slide I stole from one of George Pemberton's lectures years ago when I first started. And you can see this is a trilobite trace fossil called Rusophycus 2. We always call it a trilobite trace fossil. But this figure shows, well, horseshoe crabs could make a similar trace. Snails could make a similar trace, and polychaetes like Aphrodite could make a similar trace. So this is maybe the 95% solution, but it's not the 100% solution. And the one thing we never mentioned is that one animal can make several traces. So one type of animal may well go around making houses to live in, like this trace called Silenicnus, walking tracks called Diplecnides, and hoops called Fabrina, and then sediment balls that I think remain unnamed, but I'm not sure. Trace fossils, as we alluded to before, are behaviors. And that's how we use trace fossils to interpret sedimentary environments. Behaviors are basically survival efforts in a sedimentary environment. So if you interpret the trace fossil as a behavior, you have the potential of interpreting the environmental challenges that the animal faced in living in that sedimentary environment. For example, were the sedimentation rates high, low? Were they sporadic? Were there compromises in the chemistry of the water, like salinity and oxygen, and the type of food resource it had and how it exploited it? Because if you can imagine the end members, if the food is settling to the sea floor and becoming buried in the sediment, well, then you need to be able to deposit feed to take advantage of that sediment. But food being stored at the sediment water interface and passing into the sedimentary record is part of the sedimentary environment that reveals that you have low energies and a proximal source of food, so the food's coming in and settling, so you can take parts of the hydraulic setting apart. Whereas filter feeding means that the food's almost always in suspension and you need persistent agitation in the water. And so we link those behaviors to the sedimentary environment. This diagram goes back to Hanschel in the 50s, and you can see that there's a lot of behaviors that are referred to by kind of Greek names, I guess we'll call them. Paskychnia for grazing, agrychnia for farming. It's always a surprise to think that a little teeny invertebrate animal is farming, but you get them into deep environments where the food is otherwise poor, not a huge food supply. The animals can grow bacteria in these elaborate labyrinthine tunnels and go back through the burrow once a week and eat these bacteria that are growing there. So they really are farming. And what's interesting about these farming structures is they even look like farming structures. There's the similarity to some of these in a plowed field is actually stunning. We have tracks for running away, moving, looking for things, burrows for living in and hiding, burrows for going up and down if the sediment is grading or degrading or going back and forth and so on. Originally, this diagram back in the 50s had about half of these behaviors on it. And over time, we've added several behaviors. And this diagram is from the most recent one I know of, which we're just publishing in a paper in a new textbook on sedimentary environments, the fourth edition, third or fourth, I can't remember, of Redding's sedimentary environments. This behavioral aspect can't be understated. And in fact, the idea of these being behaviors goes back to the 40s and 30s in Germany, where researchers at the Senckenberg Institute were actually referred to trace fossils as fossil psychology, right? Like they literally viewed them as fossil psychologies that they could interpret. And why did that become so important in the ichnological landscape? Was Zeilacher, who was working in Germany, established the ichnofacies. And the ichnofacies was a recognition that these burrows could be united by their behaviors. And these assemblages were revealing behaviors about the sedimentary environment that helped you interpret things like depositional energies, food resource availability, substrate cohesiveness, and even other parameters like oxygen and salinity. And so this landscape that Zeilacher proposed, and he originally just proposed back in the 50s, this Skelethos cruziana zoophycus, which actually came later, but that's okay, and Nereides, right? So this was the landscape of ichnofacies back in the 50s and 60s, largely ignored in Europe and North America otherwise, until it was published in English in 1967 in Scientific American, which seems like an odd place for it to show up. But if you look through the history of Scientific American, every so often they got interested in esoteric paleontological stories like ichnofacies. And then these other ichnofacies, I mean, now there's like 20 ichnofacies, right? There's a whole bunch of ones for continental settings, and there's a bunch more for lacustrine and brackish water settings. And I'll be going over some of those in my presentation tomorrow. But you can take this another step. If you look at size and diversity of trace fossils, you can start to get a sense of how much stress an environment has latent within that ichnofacies. If you thought of an ichnofacies at its whole bloom, we would normally be thinking of that as fully marine salinities with full oxygenation. That's how you get the best expression of these ichnofacies. And then as you introduce chemical stress like lowered or fluctuating salinity or lowered or fluctuating oxygenation into a sedimentary environment, that drives the diversity of ichnogenera that you observe down substantially. and it drives the diameter of burrows down substantially. That means fewer animals can make trace fossils, so you get a less diverse assemblage, and only smaller animals can handle very reduced or fluctuating salinity or low O2. In the case of O2, the reason is obvious. You have not enough oxygen to support the metabolism of activities of large animals. In the case of salinity, because so many animals that live in low salinity settings are osmoregulators or osmoregulators, they have to work really hard to persist in these low salinity settings. So yeah, what's the difference between these? Well, if you were looking at the Cruziana ichnofacies, which it doesn't matter which that is, but the shelfal setting, the inner shelf, you would see that trace fossil assemblage, a good healthy one, 20 to 30 trace fossils types. And the largest diameter of those might be 20 or 25 millimeters in its diameter, right? So those are big. Okay, so we sometimes just multiply those together as a proxy for the overall health of the environment. So, 25 times 20, let's call it 500 is our size diversity index. You go over here to the inner estuary of brackish water setting, let's say, and your diversity will be 1 or 2 types of trace fossils, that's all. And the diameter of the burrows probably will be less than 3 millimeters. So your size diversity index is suddenly 6 instead of 500. And there's a continuum, obviously, throughout this. And here's an example. This is from the Jurassic Chandelat Formation in Germany. I worked on this about 13 years ago and never really published it, but it's okay. It has these beautiful examples of marine expressions like this. And you won't be able to recognize all the trace fossil types here, but this is one trace fossil type that we call that. This is in core now, and that width is 10 centimeters, just so you know. This is Astrosoma. And then these white dots here, that's another type of trace fossil that actually is some little branching trace fossil called chondrites. There's these little black flecks down here, which are phycosiphon. There's phycosiphon here and there. So there's a few trace fossils. There's phycosiphon. There's planalides. At least with a bit of work, we can find 7 or 8 types of trace fossils here, with the largest being about 20 millimeters in diameter. Whereas let's look at the same core, but in low oxygen interval, and you can see that now, yeah, it's fully bioturbated, just like the last one. But what's different? You don't have any big trace fossils at all. The trace fossil assemblage is not very distinctive, and the only things I can make out here really are these black blebs, which are Cosmoraphine. We don't need to get into the ichnotaxonomy of it now. And you have these other little blobs that we would refer to as planalides, probably. And that doesn't matter. With an overall bioturbate fabric that is hard to interpret. So it's pretty clear that, and this is a low oxygen setting. And if you go back here, that's probably more or less a fully marine expression of the trace fossils. If we wanted to, and I'll just show you this, don't get too blown away, don't get too mesmerized, don't get too lost in the diagram. I'm going to show you, oh, this isn't it yet, it's the one after this. Yeah, because the other thing we measure is bioturbation intensity, which sounds kind of obvious, right? Bioturbation intensity wasn't a thing to measure in sedimentary rocks until the late 60s, when another German researcher, Hans Reinecke, invented a system that assessed how thoroughly a sediment was disrupted by burrows. And this roughly follows Reinecke's scheme back from 67. And why was he doing this? He did this in the context of a paper, a research study that he was ascribing different bioturbation levels or intensities to different sedimentary environments. A delta would have this range and this distributions and so on. Why did that work? Well, if you really think about it, one thing we know about the end members here is to get a high bioturbation intensity, you pretty much have to have low sedimentation rates. If the sediment is accumulating rapidly, you cannot achieve this level of biogenic churning. So this is like a first order principle of ichnology. And if there's no bioturbation in it, we have two choices to interpret from. And actually either can be right. One is that the sedimentation rates are so high that they are not burrowed at all, which is entirely a possibility. Or there were just no animals living there. In other words, there was so much energy in the sedimentary environment that colonization wouldn't be possible. We get around that in a variety of ways. But if you bear with me, yeah, I think that recording bioturbation intensity really helps us backtrack to the sedimentological dynamic of the sedimentary environment. So we view it as a really important thing to measure. So there's things we can measure here. There's ichnological types of trace fossils. There's size and diversity. There's bioturbation intensity, and maybe there's a few other things, but that is going to really end up being like, this is the workflow. Identify the trace fossil assemblage, estimate their relative abundance, evaluate the diversity, interpret behaviors, measure the degree of bioturbation, compare the trace sizes, and look at the sedimentology. And to this integration, we can start to look at overall distributions in sedimentary rocks. And those overall distributions should tell us where the main perturbations and proliferations of bioturbation in the sedimentary record are and what they mean to us. And so I'll show you a case study from the Second White Specks in Alberta, which is just a single core study, but where we collected all this data in 25 centimeter bins, right? It's a lot of data. You know, normally you can log 100 meters a core in about 2 days, right? And if you log it in this detail, poor Sarah Biddle, who's working on her, just finished her PhD and started a postdoc down at UT Austin. But she was super diligent and measured all this data. It took 6 weeks, right? That's how long it took to make this data set. So suddenly the myth that doing ichnology is cheap isn't really true anymore, because time is money too. But look at all the data that was collected. Here's some lithological data. These are trace fossil types and their occurrence. And this is the diversity of ichnogenera observed. This is the size of trace fossils observed. That's the largest trace fossil in any bin. That's what that is. And then this is that size diversity index, which is just these two multiplied together. And here's the bioturbation intensity, right? And if the Second White Specks is interesting because it kind of spans what, and I was talking to Bruce about this a few years ago over the phone. He actually kind of got me interested in the Second White Specks, but I was going to look at this core at the time anyway. And this here is covering these oceanic anoxic events, right? Which are, well, we don't know how global they are, but let's call them transatlantic anoxic events, where at least they're not really anoxic everywhere you look, but oxygen drops off. And look at the ichnological data at some of these anoxic events. The SDI drops off, the maximum diameter drops off, and so on. And here's the lowest, what we would proxy to the lowest dissolved oxygen is actually probably above the oceanic anoxic event, but the data patterns are showing us something. And that's all I mean to tell you, is you can collect this really simple data that observationally you don't really even need to be an ichnologist to collect, and you can make something of it, okay? Now, I thought it would be fun, rather than boring you with the basics of ichnology, to spend at least a few minutes on my favorite case studies, if you will. And these are kind of the derivative. These are 3 or 4 slides from 3 or 4 of my favorite papers. Okay, that's all they are. This paper just came out 2 years ago, and it actually focuses on the Cretaceous Wapiti Formation in Alberta. And the Wapiti Formation is kind of distributed in this orientation because the edge of the Western Canada Sedimentary Basin goes like this. And we're looking at a dinosaur site, which is where a lot of quarrying near Grand Prairie, Alberta was. And a friend who's the paleontologist, he studies dinosaurs really at the site, bought us several blocks. And the blocks had these really neat features on them that are kind of elongate. They seem to have a ridge on them. And if you look really carefully, you even get a sense that those ridges are have kind of these lineations on them, although it's hard to say. And if you really use your imagination, you actually get the sense that there's some preferred orientations on there. There's orientations following this way, and there's orientations following that way. And if you had a really good eye, and I imagine that Guy or Alina might get the sense of it, although the scale and resolution of this photo is not quite good enough. There's actually parting lineations along this surface. And so that there's actually a record of the current directionality and the fact that there was a rapid flow. So we had this data available to us, and the features themselves are strongly asymmetric. This is one of the close-ups of the feature here, and you can see it almost looks like those roches montagnais, you know, those glacial landforms that are swept by glaciers. And then you got to remember, we were looking at the base of a bed. So if you wanted to, that's really how they look, right? So whatever this was, at first we thought, well, it could be tool marks going into this position of this deposit was in the overbank proximal to a channel, right? So it's kind of an overbank deposit. And we know it's a flood deposit because you have that sharp erosional surface there, the parting lineation, and then there's modest rhizoturbation after that, like plant. So it's a good overbank kind of deposit. And it appears to be a bit of a flood deposit, if a minor flood. And again, you can kind of see what these striations are. And so the more we looked at these, and then especially when you collect their orientation data, what I alluded to, the blue line shows the orientation of the current flow direction, which is 334 degrees. And then all of these features here are, this shows the Rose diagram, shows the orientation of these trace fossils, all of these shapes here. And you can see that for the most part, they have a mode that's perpendicular to the current and a mode that's parallel to the current, right? And so we took that as kind of the final clue that these are escape tracks of an animal that got caught in a flood in the overbank. The current water came up and the animal had to swim out of trouble. And you'll know from swimming in a current, there's only two ways in a current. There's with the current or against the current, and there's across the current. And these are in fact really similar in shape and size to the feet of Cretaceous turtles that are documented from northwestern Alberta. And so the trace maker was probably one of these guys, something like a red ear slider, which has feet really similar to the Cretaceous turtles that existed back in Alberta back then. And it was getting caught up in a rising water situation where it had to start escaping from the stream. If I had a picture of it in a real flood, I would put it over in the overbank where the stream just spilling over the overbank and the turtle going, I better get to higher ground or something like that. That's fun study one. And fun study two, I don't know if this is a fun study, but this was eye-opening to me because it showed a use of technology, like you can do something for 20 years and still not realize what you're doing kind of thing. And that's what this was. I'd worked on trace fossils a long time, and this was a core study from the McMurray Formation, strictly facies mapping. We weren't really doing it. But the more I looked at the core, the more I realized, and this had kind of developed over years, that there were two types of this heterolithic bedding. Now, for some of you, you might not realize in the McMurray Formation what that heterolithic bedding really shows. And this is called inclined heterolithic stratification. You can't tell in core, but it is inclined. It has like an 8-degree dip or something like that, right? So it always is like that. And these are pretty well interpreted as the migration of a point bar in a fluvial tidal system, right? And so that point bar is migrating. This is an outcrop of McMurray Formation. That actually shows barely dipping examples of inclined heterolithic stratification. And what you'll notice is there's a distribution of bioturbation in the different lithosomes. And after working on this for about 15 years, I realized that there's two types of bioturbation in these inclined beds. I'd been working on the McMurray since 2003, and this is now like 2018. And I'm like, well, there's examples where the mud lithosome is burrowed and the sand is cross-bedded. And there's examples where the sand lithosome is burrowed and the mud is massive appearing and looks like a fluid mud. So we have two very different types of deposits here. And for those of you who are not sedimentologically inclined, how would we interpret this? Well, this is burrowed. So this had all season for animals to live in it. That's the low energy time of year. It's the summer or whatever you want to call it. And in the summer, mud's accumulating, not so much sand, and animals are colonizing there. And in the winter, if you will, the stormy season, the sand is being deposited. You can tell by the cross-bedding there that there's a substantial current energy delivering that sand to the sedimentary environment. And so this is showing us that the sand is the flood deposit and the mud is the quiescent summer deposit. We'll just use summer and winter as our end members, even though it's not necessarily true. Over here, just the opposite has to be true. Sand is coming into the sedimentary environment when the storms are present, and mud is coming into the sedimentary environment when the storms are present. In other words, you have the river in full flood, and instead of bringing sand immediately to that depositional area, you're bringing mud. And does that happen in estuaries? Oh yeah, for sure. You know, one of the things you know if you work in estuaries is rivers come into the estuary, and where they meet the salt water, you get a zone where the flocculation of muds, which is caused by the change in the electrostatic components of salt water, they change the way clay molecules, particles stick together, and you get larger aggregate grains sticking together where you change the salinity. And so when fluvial discharge is low, you get mud being deposited closer to the river, and where fluvial discharge is high, you shift that mud belt, right? And this is the key. If you're at location A, then the high energy lithosome is the sand, and the nice good weather lithosome is the bioturbated mud. But if you're in more distal positions in the estuary, closer to the ocean, the massive muds are the high energy lithosome, and the bioturbated sands are the low energy lithosome. Super cool. And we published this in Sedimentology. I actually submitted it to Nature, but you know them. There's like, where's your geochemistry? It's a good question. This is great though, right? You know, the idea that you can actually use a biological sedimentological distribution to literally refine your position in an estuary within 30 kilometers is a fantastic notion, right? It really is. To think you can look at a deposit 100 million years old, which the McMurray is, and do this with it. So that's just a great example of what ichnology can do. And our last example, which is my favorite one, is from Willapa Bay. And this is, remember, where I did my PhD. So a couple of the photos are a little fuzzy in this, okay? That goes back to the 90s. But we're in a location up at North Cove with a set of sand outcrops that actually are Pleistocene age inlet deposits, and the modern inlet here, and the Pleistocene inlet is here. And you can see there's actually like 30 meters of cross-bedded and locally bioturbated sand in it. I'd worked on this for quite a few Well, I'd say about 2 weeks. I was just measuring the sand pile. And then I started to realize that this is the most fuzzy picture you'll get in the whole presentation. We do better than that in 2 slides. Dave Bechtel, a friend who visited me out there. And they had these weird pits, and they were actually intersecting these bizarre columns, right? So these are biogenic sedimentary structures. I became convinced of it, but I couldn't understand what I was looking at fully. So you don't see the columns very well here, but there's one there. That's one of the columns. They were all over the place. There are literally hundreds of these, each type, the columns and the bowls in each outcrop. And you can kind of get a sense of their distribution there, but it's better to look. Oh, okay, cut a long story short. I thought I knew what the vertical columns were. This is a trace fossil made by a bivalve called Siphonichnus, where faunalized bivalves live in the sediment literally for decades, and they can move up and down as the sediment degrades. And so you get these reworked columns in the rock record. So I felt good about that interpretation because it really resonated super well with other trace fossils I'd seen at other locations. But the bowls, and that's a core that I'd taken literally the same year I was out there working on this with a bivalve called Maya arenaria, living down about 35 centimeters below the sediment water interface. So they reach up there with their siphon, and that's where they get their food, right? These guys are mostly filter feeding, but there are bivalves that deposit feed this way too. They have very flexible siphons, and they can go up to the sediment water interface and literally drag food into their siphon. So and what's the advantage of this behavior? Well, you're pretty safe from predation, right? So I thought I had a good model for the columns. And what I didn't understand was what were these bowls, right? So here's one of the bowls right here a little bit. And here's a better one. That's the best one. And in this bowl, you'll start to get a sense. Something that occurred to me fairly soon was that there are coarser grains here and coarser grains here. And there was always this channel up the middle where there were coarser grains being pulled into the middle and upwards. And so it became clear to me that this was the type of feeding structure that was hydraulic in some way. And there were candidates. So it could either be a whirlpool feature, a hydraulic feature made by currents, but they were all over the outcrop. Or it could be a biogenic sedimentary structure. And are there animals that dig hydraulically to eat? And the answer is absolutely. There's rays. They jet into the sediment to make bowls and bring the food up. There's other animals that dig in these sediments, in these settings, like even raccoons in intertidal flats. But I couldn't think of what would actually be jetting enough to lift and move gravel. And so, in fact, I should have put a picture from my field book there because I literally drew a woolly mammoth with its nose actually digging and jetting in the sediment, which, as it turns out, probably wasn't wrong because that summer I read about walruses and the fact that walruses were using jetted water from their big mouths to jet into the sediment water interface down into the gravels. And in fact, a National Geographic paper came out at exactly the same time was trying to interpret these that showed all of these walrus feeding traces. And guess what they found as the stomach contents in walruses that they caught? Bivalve siphons in all of them. They were exclusively jetting for bivalves. And so that's when I decided that was the ichnological interpretation, right? So the walrus uses its vibrissae, and this is how they do it in the modern. They have these large whiskers like a cat does, and they go along the bottom until they get a little movement from a bivalve, and then they jet into it and they pull it up into their mouth. They get their mouth around it and they suck the meat out, and then they discard the shell. And so they can really suck and really pull really hard to the point where the same summer I was researching, I was I called up a couple zoos down on the West Coast and I asked them what they thought about walruses. And they had an example where an walrus was living in one of the SeaWorld places and they had to clean its tank or make changes to its tank. So they put it in with the seal tank for a couple days. And for some reason, it got angry with the seal and put its mouth on the head of the seal and sucked its skull cap and brains out, right? Literally. And then I'm like, yeah, that's our guy. Yeah, for sure. So that's the story of the walrus traces. Again, I submitted this to Science, but they weren't too impressed. And it ended up in Paleo. So this came out much later when National Geographic managed to get video, and you can see this on YouTube. You can see this video of the walruses actually jetting into the substrate, going for bivalves, which is almost what they entirely eat. It's pretty amazing. So yeah, this is probably my favorite ichnological story. Yeah. So ichnology, well, it's how animals interact with sediment. There's several reasons why we study them, but I think the best reason to give you is we interpret sedimentary rocks much, much better if we study the ichnology. And, I hope where you've arrived at the end of this talk is at least that, when you're at the beach and you see an animal living in it, or if you see a rock, and you often see rock with bioturbation in it, you'll realize that, there's hidden secrets in that rock that you probably weren't thinking about. And so I'd say thank you for your invitation to come here. It's been an honor, and for your attendance.
Like a conversation over some.
Oh, I didn't realize how, wow, that's a little longer than I normally go. I obviously really like y'all because I don't normally do that. Yeah. Sorry. Yeah, sure.
Can you, given the phenomenal book control, you actually able to demonstrate that we can follow one layer from science to another?
The core control might do it, but the certainty of correlating beds is really